Impact of food toxins on the disruption of cellular processes. Food toxins pose a significant threat to health and well-being by disrupting essential cellular processes. These disruptions can lead to DNA damage, inhibit repair mechanisms, and cause oxidative stress and inflammation, all of which are linked to a variety of chronic diseases, including cancer, cardiovascular diseases, and neurodegenerative disorders. Understanding the mechanisms by which food toxins affect cellular health is crucial for developing strategies to mitigate their impact and promote overall well-being. DNA damage and repair inhibition. One of the most severe impacts of food toxins is their ability to cause DNA damage and inhibit the body's natural repair mechanisms. DNA damage can occur through various pathways, including direct interaction with the DNA molecule or through the generation of reactive intermediates that harm genetic material. Persistent DNA damage can lead to mutations, which, if not properly repaired, can result in cancer and other genetic disorders. Heavy metals such as lead, mercury, and cadmium are known to cause significant DNA damage. These metals can bind to DNA and interfere with replication and transcription processes, leading to breaks and errors in the DNA sequence. Additionally, they can disrupt the activity of DNA repair enzymes, preventing the correction of these errors. For instance, cadmium has been shown to inhibit the DNA repair process by interfering with the repair enzyme's active sites, leading to an accumulation of DNA damage over time. Pesticides, especially organophosphates, also contribute to DNA damage. These chemicals can form adducts with DNA, resulting in structural changes that impede replication and repair. The inhibition of DNA repair enzymes by pesticides further exacerbates the accumulation of genetic mutations, increasing the risk of cancer and other chronic diseases. Oxidative stress and inflammation. Oxidative stress and inflammation are closely linked processes that play a central role in the pathogenesis of many chronic diseases. Food toxins can induce oxidative stress by generating reactive oxygen species, ROS, that overwhelm the body's antioxidant defenses. This imbalance leads to cellular damage, inflammation, and a cascade of harmful effects on overall health. Reactive oxygen species, ROS, are highly reactive molecules that can damage cellular components, including lipids, proteins, and DNA. When ROS levels exceed the body's capacity to neutralize them, oxidative stress occurs. This stress can lead to lipid peroxidation, protein oxidation, and DNA damage, contributing to cellular dysfunction and disease. One prominent example of a food toxin that induces oxidative stress is bisphenol A, BPA, commonly found in plastics and food containers. BPA exposure has been linked to increased ROS production and oxidative damage in cells. This oxidative stress can trigger chronic inflammation, a key factor in the development of conditions such as atherosclerosis, diabetes, and neurodegenerative diseases. Heavy metals, such as mercury and lead, also contribute to oxidative stress. Mercury can generate ROS through its interactions with cellular components, leading to mitochondrial dysfunction and increased oxidative damage. Lead exposure results in the depletion of antioxidants like glutathione, further exacerbating oxidative stress and promoting inflammation. Inflammation is the body's natural response to injury or infection, but chronic inflammation resulting from persistent exposure to food toxins can lead to tissue damage and contribute to the development of chronic diseases. For example, chronic exposure to pesticides has been shown to activate inflammatory pathways, leading to increased levels of pro-inflammatory cytokines. These inflammatory responses can damage tissues and organs, contributing to diseases such as inflammatory bowel disease, cardiovascular disease, and cancer. Mitigating the impact of food toxins. Reducing the impact of food toxins on cellular processes requires a multifaceted approach, including regulatory measures and individual actions. Regulatory agencies must enforce stricter limits on environmental contaminants and ensure safer food production practices. Promoting the use of organic farming and reducing the use of harmful pesticides can significantly decrease exposure to these toxins. Individuals can take proactive steps to minimize their exposure to food toxins by choosing organic produce, avoiding plastic containers for food storage, and opting for BPA-free products. Consuming a diet rich in antioxidants, such as fruits and vegetables, can help bolster the body's defenses against oxidative stress and inflammation. Public education campaigns are essential to raise awareness about the risks associated with food toxins and to encourage healthier dietary choices. By understanding the sources and effects of these toxins, individuals can make informed decisions that protect their health and well-being. Conclusion: food toxins significantly disrupt cellular processes, leading to DNA damage, inhibition of repair mechanisms, oxidative stress, and inflammation. Understanding these impacts and taking preventive measures can improve health outcomes and enhance overall well-being. By addressing environmental pollutants and promoting safer practices, we can protect public health and reduce the burden of chronic diseases.